Tumor grading, what makes comparison challenging? Imagine trying to compare apples and oranges to decide which is better. That's similar to what doctors face when comparing tumor grades across different brain tumors. Tumor grading helps us understand how aggressive a tumor might be, how fast it grows, and how likely it is to spread. But making direct comparisons isn't straightforward because of several complex factors. First, different tumor types have their own grading systems. The World Health Organization uses a system that grades tumors based on specific features within each tumor type. For brain tumors, this means that a grade 1 tumor in one type might be very different from a grade 1 in another. Even though both are labeled as low grade, their behavior and prognosis can vary widely. Recent updates have standardized some parts of these systems, but the core idea remains that each tumor type has its own set of rules, making cross-comparison tricky. Second, grading relies heavily on microscopic examination by pathologists. They look at tissue samples and interpret features like how many cells are dividing, whether there is dead tissue or necrosis, and how abnormal the cells look. Since this interpretation involves human judgment, it can vary from one pathologist to another or from one institution to another. This subjectivity can lead to differences in grading, even for the same tumor. Third, advances in molecular biology have added new layers to tumor classification. We now look at genetic and molecular markers that can tell us more about how a tumor behaves. These markers sometimes define new tumor subtypes that don't fit neatly into traditional grading schemes. For example, two tumors might look similar under the microscope but have different genetic profiles that influence their aggressiveness and response to treatment. This complexity makes direct grade comparisons more difficult. Fourth, the environment around the tumor, called the microenvironment, also influences how a tumor grows. Some tumors have non-cancerous cells that help or hinder their growth. Traditional grading systems may not fully account for this complexity, which can lead to differences in how aggressive a tumor appears based on the grading criteria used. Fifth, diagnostic tools like imaging scans and biopsy techniques can also impact grading. Variations in MRI or CT scans, or differences in how tissue samples are taken, can lead to under- or overgrading. Tumors that are hard to access or small in size might not be fully represented in the tissue sample, making accurate grading more challenging. Lastly, tumors are not static. They can change over time, becoming more aggressive or recurring with higher grades. This evolution means that a tumor's grade at diagnosis might not reflect its future behavior, adding another layer of complexity when comparing tumors at different stages or times. All these factors highlight why comparing tumor grades across different brain tumors is so complicated. It's why doctors emphasize a personalized approach, considering not just the grade but also the tumor's molecular profile, location, and the patient's overall health. Accurate diagnosis and treatment planning depend on integrating multiple pieces of information, not just the grade alone. Understanding these challenges helps us appreciate the importance of multidisciplinary teams and standardized protocols to improve consistency and care in brain tumor management. Always remember, for any specific diagnosis or treatment, consulting healthcare professionals is essential to get advice tailored to individual cases.